Welcome to OpenSociety.wtf, a podcast where we explore how today's technology shapes our world, both in building a more open digital society and in creating its opposite. We talk to experts in biotech, Web3, and AI to find out how these innovations are impacting our daily lives. Today's episode features Chase Denecke, co-founder and CEO of Bootstrap Bio, and co-author of the popular Let's Run article, Significantly Enhancing Adult Intelligence with Gene Editing May Be Possible. Chase and his team's ambitious goals align with today's longevity movement, led by figures like Brian Johnson and the crypto city Prospera. But is the dream of multi-gene editing to enhance intelligence a threat to our future society? Could it be monopolized by minority, leading to a dystopian future? And beyond the dream, is this startup's vision realistic in the first place? Chase's research proposal has already caught the attention of Silicon Valley venture capitalists Balaji Srinivasan and Brian Armstrong, both well known in the cryptocurrency space. In this conversation with Chase Denecke, we explore the ethical dilemmas of gene editing for intelligence, whether it could exacerbate societal inequalities, and also dive into some of the technical aspects of multi-gene editing. So I spent some time reading your investment memo and also Let's Run blog post. But I realized in the end that the Let's Run blog post has like 90 pages. So I give up on that.
Long.
Yeah.
There is a summary of it at the bottom in one of the comments.
Mm-hmm. Okay. Yeah. So I just went through your investment proposal. And I feel quite excited reading about this. And
Cool. I'm glad you found it interesting.
yeah, so you also mentioned a lot of misconceptions people have for biology, side effects, mosaics, and also a lot of unpredictability of editing genes in multiple cells simultaneously. I found that quite interesting. It's just I don't know how you guys gonna achieve this.
Interesting. Which part? Like the, editing in the liver or in the brain or just like the entire thing?
Well, editing the brain, yeah, definitely very hard. It's like you guys targeted 2027, 2028 as the milestone for the first delivery to the brain, which is quite amazing. Considering brain is heterogeneous and you want to deliver editors to multiple cells at the same time. For me, it's like, wow. But we can go
There's actually
deeper.
Yeah, sure. I
Mhm.
mean, there's actually AAV capsids from Dino Therapeutics right now that can already deliver editors to the brain, at least in non-human primates, and they probably work on humans as well. The thing is, the efficiency is not super high, and you can't redose someone with those AAVs, and they're also super expensive. Like, it's already possible to deliver to the brain. It's just not very efficient.
Right. So, okay, you talk about misconceptions and you also talk about, you relate your experience in working as a computer scientist and also a biologist, and you kind of understand the complexities, and people shouldn't really be able to understand every gene processes to understand the human body. And I guess for GPT, or neural networks, it's a black box as well. So I guess we would start with some questions about consciousness in AI and how does it relate to human consciousness? If you
Consciousness in AI. Okay, all right.
Yeah,
Cool.
because they both are, well, black box. And do
Yeah.
you find any similarities?
Do I find any similarities? I mean, for sure, there's definitely some similarities in, the structure of like neural networks and brains. Obviously, like the structure of neural networks in general was inspired by brains. This sort of like sequential layers of neurons that are interconnected with each other. That was originally inspired by the brain. Now, there's obviously some differences. But even when it comes to the fundamental kind of like update processes of the brain versus neural networks, I think like the most well-supported theory for like how the brain actually learns is via this process called like uh, it's predictive coding, I believe. And there's a mathematical description of how predictive coding works. And I think a couple of years ago, someone showed that you can mathematically reduce back propagation. You can mathematically reduce predictive coding to back propagation. In other words, like one process is basically equivalent to the other, which I found one of the most interesting results I've heard of in computer science in a while. Because it basically suggests that whatever the brain is doing, it's basically at some level equivalent to what neural networks are doing. The way they learn is very similar. Now, I'm sure a neuroscientist would probably tell me there's major differences, but the fact that there's similarities at that level is pretty amazing to me.
You mentioned about several misconceptions in biology, and would you share with us again here, and why do you think that's causing a lot of misconceptions for people to be intimidated in such a complex system?
Right. So there's a lot of misconceptions in genetics in particular. I think that so I'll give you one simple example. If you talk to, some people in the field of genetics, they'll insist that like everything is extremely complicated in the genome and that, all genes are hopelessly tangled up with one another. And if you modify one, that you can have no idea how it will impact the rest of the genome. This is basically not true. The effects of genes on traits are genes mostly affect one trait. There's this idea called pleiotropy, which is basically one gene affecting multiple things. This definitely happens. It's present in the genome, but it's not all that common. And then, even within a single trait, like, let's say you're looking at like diabetes risk, right? Some people think that like, if you modify this one gene, it might decrease diabetes risk, but like it could have this knock-on effect on all these other genes, and like that might increase diabetes risk. This is basically not true. The effects of genes are almost entirely linear, meaning you can just add up the effect of a bunch of different genes and through that create a pretty good model of how those genes are going to affect a particular trait, like disease risk. I think there's a decent chunk of the genetics field that's basically ideologically motivated, meaning there's a large chunk of academia that doesn't like the implications of genes affecting life outcomes and, whatever that might imply. And because of this, there's a lot of, for lack of a better term, misinformation about genetics, often, promulgated by the field itself. Now, if you actually dig into the research papers, you'll find that like the data, supports a simpler conclusion usually. But the way that academics will talk about their research is different from what the data actually says. For example, they'll highlight certain findings they have. I remember attending a conference in Boston on polygenic embryo screening, which is a practice that parents do during IVF that helps them have children with like lower risk of disease. And you'd think like everyone would be in favor of this, right? Because who doesn't want, who wants a kid who has like disease? But everyone there absolutely hates the idea. And so they, all these speakers get up on stage and they like make these claims about how, oh, we can never know what these genes are doing. We can't predict what disease risk will be. The predictors aren't good enough. This won't work. Also, if it did work, it would be bad. It's just kind of like amazing to see how like deranged the field of academia has become about this topic.
So it's people don't like the idea of being editable intelligence.
It's even more profound than that. Not only do they not like the idea of editing intelligence, they don't like the idea of like picking an embryo that has a lower risk of disease. Like imagine you have 5 embryos, right? And your choice is let the embryologist pick the embryo based on how good it looks to them under the microscope, or do I pick an embryo based on how likely it is to like have a heart attack? And a bunch of academics are like, it's immoral to pick the embryo that has the lowest risk of heart attack. Literally.
So I think you mentioned another question I really want to ask. In the Let's Run blog post and in the very early introductions, you mentioned about when you grew up, you liked the book about AI, basically. And then later you encountered embryo selections. And how does the later encounter of embryo selections change your perspective about AI? And what's your kind of picture of in the future, how would human interact with AI?
Oh yeah. How did embryo selection change my views of AI? That's a good question. I'm not sure it directly changed my view of AI. Um, Frankly, I think since maybe 2016 or so, I had been somewhat concerned about the trajectory of AI, simply because the technology was obviously going to be extremely powerful. And so if we didn't manage it well, it seemed like there was potential for major disaster. Um, I'd hoped for a while that, you know, so should I explain what embryo selection is first, so like the audience can understand? IVF is a process that couples go through, often if they are having trouble conceiving, right? They're trying to have a kid and they're having trouble with it. Sometimes most parents do it because they're having trouble having a kid, but some do it because they have a genetic disease in their family that they don't want to pass on. There's different reasons people do it, but part of the IVF process involves, the mother going into a clinic, taking some hormones, and then extracting eggs, which the clinic will fertilize using the father's sperm. At the end of that process, if things go well, you'll have multiple embryos, which any one of which could potentially become a baby. Um, you know, if you're fortunate, you may have like, 5 embryos that could become a baby, right? Most people don't want 5 kids, so there's often a choice that has to be made about which embryo do you implant first, right? You have 5 to pick from. Which one do you pick? The process by which this used to be done is called morphology-based screening, which is a fancy term for an embryologist basically looks at each one under the microscope and says, nice. And then they transfer that embryo. And that's how you determine which one will become your kid. Embryo screening is this idea that, okay, instead of just looking at how nice the embryo looks, we're going to look at its genome. And we're going to use the information present in the genome to learn about its predisposition to disease and maybe other stuff as well. Right now, there's a couple companies offering embryo screening services. There's one called Genomic Prediction, and there's another one called Orchid Health. And they both just screen for disease risk. But theoretically, you could use this technology to screen for anything. Anything that has a genetic basis could be screened for. The predictors for different traits are not always equally as good. So, for example, our predictors for height are very strong, but our predictors for personality are quite weak. So, you could select for both theoretically, but you can't have as large of an impact on personality as you could on height, at least not currently. So I learned about this technology a couple years ago, and I thought like, wow, this sounds like extremely powerful. Like imagine if, a good chunk of the world's kids were born with like lower disease risk and like, maybe even like slightly increased intelligence and whatever. Take your pick of other traits that are important. So in terms of how that affects, AI, my thought was, 5 years ago that if we had enough time until someone creates AGI, the next generation of kids, that are born now that will grow up in the future might be a bit smarter, might be a bit healthier, and like perhaps they could actually kind of help guide development of this technology. Now, the effect size from embryo selection is not that strong. For example, if you're selecting just for IQ, right now, using current predictors, you could probably gain about maybe 6 IQ points at most, maybe 7 if you push it. So, a reasonable effect size, but not a huge one. So most of my focus on the way that this technology might impact AI was looking at some of these future, more powerful technologies that would kind of surpass the size of the benefit of embryo selection. You might wonder, okay, well, like, what are those technologies, and like, why might they be better than embryo selection? The core issue with embryo selection is you're selecting a sort of a random variable from a normal distribution. So, like, think of the bell curve, right? If you're like trying to, make your kid an NBA player, you might select the tallest one, right? But you're selecting the tallest one from this sort of bell curve distribution. And that means that if you have a ton of embryos to pick from, it's only a little bit better if you have than if you have like 10 embryos to pick from. So like if you have 10 embryos to pick from, you might get like 2 inches of height gain. And if you have like 1,000, you might get 3. And if you have like 1,000,000, you might get like 3.5. So the scaling laws here are terrible. You can't just have a much larger gain by having more embryos. So the question then becomes, okay, well, is there a better way to do this? And the answer is yes, of course. There's various methods that are plausible to do, but one of the simplest is just gene editing.
So disease and intelligence you mentioned.
Yeah.
For me, there are two different things. Maybe for
Yeah.
most people would maybe repelled of thinking, oh wow, we can select embryos or we can edit genes for better intelligence. For many people, it would be intimidating in some ways. But as you imagine, in your vision, AGI will happen, and humans have to be competitive enough to reach the intelligence level of the machines.
Yeah.
What's
I
your
think
model
that's
of editing intelligence?
yeah. Good. Okay, a couple of good questions there. So I guess to start off, I think that it's going to be hard for humans to compete with AGI in the long term. I think that in some ways, computer-based intelligence is just fundamentally superior to that of like biologically based intelligence. So I think it's going to be quite hard for us to like keep up with AI. In fact, I think it's going to be impossible. So I really view this technology as kind of like a bridge, sort of bridge to a future where we can actually control these extremely powerful machines that we're creating. Let's see. And then in terms of like the ethics of enhancing intelligence, I mean, there's definitely debate on this. I've never completely understood the view that we should like decrease disease but not increase intelligence. I mean, some of the same motivations that make us try to, decrease disease risk are the same ones that make us try to, help our kids be smarter. For example, parents try to avoid they try to avoid exposing their children to like leaded paint, right? Because leaded paint decreases IQ and causes neurological issues. Well, if, avoiding decreases to IQ is good, then maybe increasing IQ should also be good. So I think it's pretty consistent with the values that we already express.
So logically, you think it has no problem of increasing intelligence as opposed to decreasing intelligence for the sake of better health. So how do you convince people that this is actually not intimidating? I think people are just scared of not knowing what's going to happen. If everyone's can be selected to be smarter, and usually it's only a few people can do that. I don't think everyone can afford gene editing.
Yeah, this is definitely like one of the issues, right? How do you make sure that like there's equal access and this doesn't just become a tool for like rich people to become even more powerful? I think that's definitely a concern.
Yeah.
I think in my view, the answer is basically just like make the technology really cheap and make it broadly accessible. If you can solve the cost problem, you're 90% of the way there. The last 10% is just like, if the technology is really powerful and it's cheap, then you can just convince governments to subsidize it. Because if your tax base is a lot more talented, you're going to earn more tax revenue, and therefore it makes sense to subsidize, access to this technology. There's other kind of simpler things that we can do in the short term. For example, there's a large difference in the quality of IVF clinics. The top 25% of IVF clinics have a live birth rate per egg retrieval about twice that of the bottom 25%. very few patients understand this, and they don't know which clinics are better. So really simple things like directing patients to better clinics could really help a lot. Another company I started actually is working on that as well. So yeah, I think long term the answer is just like make sure everyone has access and make sure cost is not a barrier.
Yeah, I agree. So I know Bootstrap Bio is building gene editing, multi-gene editing tools to enhance, I think, first disease and second intelligence in the future, maybe. So
Yeah. I mean,
from your
intelligence is it's a ways down the road. And obviously right now we're focused on just
diseases.
liver disease is our first target. Um, so it's going to be a while before we think seriously about enhancing intelligence, but it's possible down the road.
Yeah, so you had a background in genomics and selecting embryos, maybe not the morphological selection, but more of genomic selections. And
Right.
then you moved to gene editing, as you said, which is superior than more scalable, not superior, more scalable than selecting
Right.
10, 20, 30 embryos for creating this bell chart and selecting the highest, the best ones. So when was the moment you realized, okay, well, gene editing is more scalable and it's actually achievable in the time frame you have now? And in the investment memo, you mentioned about your CTO is the inventor of CRISPR-DAP, array
Yeah.
technique, which allows multi-gene editing in multiple cells as simultaneously. So that's actually a quite interesting thing. And what's your story of bumping into this, and which kind of change your idea of, oh wow, we should probably start doing gene editing now instead of embryo selections?
So I wouldn't say that we should necessarily start doing gene editing now instead of embryo selection, because gene editing for embryos is still that still hasn't been done even once. Well, it's been done once by this Chinese scientist named He Jiankui,
Mm-hmm.
but people got very upset with him, and the technique that he used is not it's not that powerful, and it's riskier than what could be done. Um, we can talk more about that later if you'd like. Um,
Yeah, sure.
so the focus I had was basically on adult gene editing. So I mean, gene editing has been around for a while, and like gene therapy to modify genes in adults has been here for a little bit now. The first FDA approved CRISPR-based gene therapy was approved in I think December of last year. Um, but the techniques, all the therapies that are like currently either approved or in the works, in clinical trials, are basically monogenic gene therapies. So a single gene is being targeted. And if you look at one of my main takeaways from working at genomic prediction on embryo selection was that most conditions are involve more than one gene. Breast cancer risk involves more than one gene, heart attack risk involves more than one, diabetes, Alzheimer's, all these things involve multiple genes. So if you want to target any of these conditions with gene therapy, you have to go after more than one gene. The question was just always like, could that be done? And for a long time, I thought the answer was no. And there's a lot of reasons for this, but one of the biggest ones is like, okay, how do you actually get the editors to the cells in the first place? And if you can get them to the cells, how do you know that they'll edit the right gene? And what if some cells get edits and some don't? There's a lot of these technical questions, and previously I thought that these basically would make this impossible. Um, Last summer, my friend and now co-founder Ben sent me a message on Discord, basically saying, "Hey, uh, like gene editors have gotten so good that we might actually, be able to do editing in adults." And I was skeptical, but the idea seemed promising enough to be worth looking into. So Ben and I started this big research project to try to see, like, okay, if there is a hard blocker here. This is in fact impossible. Why isn't it possible? We wanted to answer at least why it was impossible. And after a couple months of looking into this, I started to think like, wait, actually, I think this is actually possible. And like, what's more, no one's working on it. All the companies working in gene therapy are doing single disease mutations, right? They're doing like a sickle cell or like cystic fibrosis or other conditions like that. So we basically wrote up our findings into this big blog post and submitted it online to this website called Less Wrong. And the post just absolutely exploded and had all this interest from investors. People wanted to know like if we were going to start a company. And at the time, we really were just looking for an investment, like a grant to study this more. But there was so much interest from investors that we figured, we can just start this in the context of a company. And so we did. You also asked about Elijah Yuan. We kind of met him and what his background is. Do you want me to get into like how we met him or like how his technology works or what?
We can go with whatever you want to say in the most comfortable way. Yeah.
So Ben and I don't have a like lab background, and if you want to actually make a therapy, you have to have someone who like has serious experience in a lab. So our first priority as a company was basically find the head of our lab, and the question was like, okay, how do you find someone like that? Because it's very hard to find someone with a very specialized skill set to do this type of thing. We had read a lot of papers about gene editing and gene therapy, and very few of the papers we read had actually discussed doing multiple edits at once. But one particular paper published in 2022 had, so far as I could tell, the record for the most number of gene edits conducted simultaneously in one cell. And that paper was unusual in a number of ways. One, they did like 31 cells in one cell, but second of all, there was only 2 authors on the paper. And that's quite unusual. Most of these papers have, maybe 5 to 10 authors. And when I looked at each one of the authors, it was clear to me that like one of them was the PI, basically the professor, who like usually doesn't do that much direct work on a paper. So I was like, wow, like this whole paper was done by this one person. And the natural next question is like, who is this guy? His name listed on the paper was Chi-Chen Yuan, and I tried to look this guy up, and I couldn't find anything about him online. There was no email address. He had no Twitter profile. He had no LinkedIn. I couldn't find anything. So it's like, man, I want to talk to this guy, but like, how? How do I get in contact? I couldn't figure it out. So eventually what I did was I looked at his research collaborators on ResearchGate, and one of them had uploaded a profile picture to his ResearchGate profile. So I figured like, okay, this guy probably uses social media. Maybe I can find him on another platform and might contact him. So I looked for him on LinkedIn and I found him. It's like, his profile picture looked the same as it did on ResearchGate. So I was like, okay, I'm going to reach out to this guy and ask him if he can put me in contact with Chi-Chen Yuan. And amazingly, he responded. And I was like, hey, I'm running this gene editing company. We're really interested in Chi-Chen's work. And it's like, okay, let me give you his email. So he gave me his email. And, what I discovered is that all of his social media profiles were under Elijah Yuan, but his papers were under Chi-Chen Yuan, which is why I couldn't find him before.
That's why.
But we scheduled a call with him, and like it was amazing. He had the exact skill set that we were looking for, and he is very well qualified to work on this stuff. So he became our chief science officer.
Yeah. So what happened during the several months period where you did this research project together with Ben and found out it's feasible to edit multiple genes, multiple cells in living organisms?
So, okay, I'll take you back to my list of things that I thought would make this impossible. So question 1 was like, how do you actually get the gene editors into the cells in the first place? I was like, okay, this, especially when it comes to the brain, there's like 200 billion brain cells. getting editors into 200 billion cells, I mean, like completely impossible. But then I started looking into like research that had been done, and I found this particular paper where the authors had like managed to transfect. They had made edits successfully in like 60% of the neurons in a mouse brain, and I was like, whoa, like 60%, like that's actually pretty good. Um, and I looked into other papers that targeted different organs, and they often had even higher efficiencies. For example, there were some papers where they were able to get gene edits done successfully in like 80% of liver cells. So just kind of immediately I was like, okay, wait, actually this might be possible. Now for the brain, it's extra challenging because you have this thing called the blood-brain barrier, which like separates the bloodstream from the brain, and getting large compounds across that is very tricky. This is tricky for like the pharmaceutical industry as well. There's ways around that, but so far as I can tell, no one has like fully solved that problem yet. This is one of the reasons the company, even though the post talks about stuff on the brain, like maybe Alzheimer's, we ultimately decided to pivot and focus on like liver disease instead, because it's quite easy to get editors to liver cells, but it's much more challenging to get them to the brain. So yeah, problem one, delivery, like potentially solvable, definitely solvable for other organs besides the brain. Problem two, how do you know the gene editors are going to edit the right thing? Back when CRISPR first came out in 2012, the rate at which it edited the wrong gene was very high. Original 2012 style CRISPR, it might make a correct edit in like a few percent of cells, maybe like 2 or 3%, I think. But since that original tool came out, there's been a couple major innovations, mostly coming out of the Liu Lab at the Broad Institute. One of them is called a base editor, and the other one is called a prime editor. I guess I'll skip over base editors for now because we're probably not going to be using base editors that much. But prime editors are super interesting. The biggest difference between them and old school 2012 style CRISPR is prime editors they only break one strand of the DNA. So like normal old school CRISPR, it like chops the DNA in half, like karate chop. And this causes a lot of problems because the repair process that stitches the DNA back together is really messy. It's like sloppy. But prime editors don't do that. Prime editors cut only a single strand of DNA, and then I can explain part of the process. Basically, they cut one strand of DNA, they pull up like a flap, and then they add bases onto the end of that flap. And then there's a complicated process whereby the DNA gets repaired, but the end result is like you have both strands of DNA changed, but the DNA was never broken in half. This is really good because it means you don't deal with this sloppy repair process that normally fixes double-stranded breaks. So the rate at which you could, the ratio of like successful edits to errors for prime editors was way, way higher than old school CRISPR, like, several hundred times better. And when I learned that, I was like, whoa, like this is so much better than old school CRISPR. Like this might actually be able to work. So that was update number 2. I also had a question about like, okay, well, what if some of the cells get edits and some of them don't? And there, we started looking at like, okay, by the time you're like age 40, how many different random mutations do your cells have? And the answer varies a lot depending on cell type, but in general, it's somewhere between like a few thousand and like a few tens of thousands of like random errors that your cells have accrued. So, just via the natural aging process, your cells already accrue a lot of differences between them in their DNA. So if they're robust enough to survive that, and like, most people don't like, most people are functional in their 40s, then perhaps having different cells with different genes is not actually going to be that big of a deal. There's some more things to make this complicated, like some of the edits, some of the random mutations happen in places that don't really matter in the DNA. So there's some more complicated analysis that has to be done. But when we did that complicated analysis, we figured out like, okay, actually, this probably is not going to be a big deal. So three problems basically checked off, right? There were some other issues that we were concerned about. One of them is like how the immune system reacts to the editor proteins, or like the DNA that encodes them. Those are still somewhat of a potential concern, but there's different strategies you can use to potentially get around that problem. Several of which were kind of investigating. So that seemed like it might be a problem, but it wasn't necessarily a deal breaker like we thought it might be. So yeah, just one by one as we wrote this post, like it's like bowling pins falling over. It's like, okay, this is not a deal breaker. This is not a deal breaker. This one is hard but like solvable. And slowly just kind of occurred to me, I was like, wait, there's nothing actually stopping us from doing this. It's all like all the pieces are there, but no one has put them together yet.
I really like the way you think and tick box in terms of solving the three problems. The first one's the delivery problem, so you guys probably choose liver to start instead of the brain. The second one, the editors, you have the prime editors as opposed to base editor, which is, I guess, less efficient in that way. And the third one is, I forgot what the third one is, but my point is like I really like the way you
Mosaicism,
think.
yeah.
What if the side effects, and the varieties of editing. Because you were a computer science before, and you worked
Right.
as a full stack web developer for many years. Do you find any difference in terms of thinking between you and Yuan, and also Ben? Because you are more like,
Good
this
question.
is logical, so we can do this, and then we tick the box. And I think for biologists, people who probably they did their degree in undergrad, they will probably be intimidated or not intimidated, but like they know how complex the system is. Maybe it might pull back from like putting different components together, that kind of thinking. But you obviously have this background and you did some genomics along the way. So do you find any differences between you and Ben and Yuan in terms of thinking?
So Ben and I both actually have a computer science background, so I think we, in some ways, think pretty similarly.
Mm-hmm.
In terms of Elijah, Yuan, I mean, we definitely think differently, but it's hard to attribute differences in our thinking styles to our respective academic backgrounds. I think there's definitely a lot of differences in biology versus computer science. Biology is just so much more like sloppy, for lack of a better term, and like unknown. With a computer program, you can like inspect the code, you can like formally verify that stuff should work. You can like guarantee runtime. Uh, there's all kinds of like really nice properties that like computer code has, and biology just doesn't have that. Like it's very stochastic, it's very random. There's a lot of like complicated feedback processes. So it's in that sense, it's actually harder than computer science. So I think if there's a way that I think the biggest.
Mm-hmm.
Go ahead.
I think for you guys, for computer science students or people who study that, you guys are primed in terms of algorithmic thinking. I don't really have that skill yet. I'm not sure if Yuan has that skill, but Yuan's probably more non-linear thinking. But you mentioned that like genetics sometimes it's linear. So he must see the beauty of logical and the sloppiness in the system.
Beauty of sloppiness. I've never heard that term before, but I like it.
Well, I just made up. It's the sloppiness and also linearity, basically linear, non-linear within the biological system that we have observed. Yeah, let's break down. Oh, you were gonna say something, right? So I just cut your.
I was actually going to bring up the exact example that you brought up. I think one of the biggest differences I've seen in sort of the way that I think about bio versus the way that kind of people in the field think about it, it's people in the field assume that it must be complicated, that somehow everything must be tied together in this hopelessly complex web. And I think a big reason why they think that way is because they see cellular processes, like metabolism, and they see how complicated it is and how many feedback loops there are. But from a CS perspective, you can just like look at, you can look at how much of the variance in a trait is explained by linear effects and just be like, wow, it's like, over 80% of the effects are purely linear. And if they're purely linear, they're easy to predict. And it doesn't matter what the underlying biology is. This linear model actually describes, the underlying complexity quite well. So just like taking that idea seriously and assuming that it actually could be that simple. I think that's something a lot of biologists just don't do.
Yeah, there are a lot of conserved features, in which can convert into what you're saying about 80% of them are linear. Yeah. So for the CRISPR editing, let's break down to 3 different ways. So you edit single gene, just within the gene itself, and you also you edit a single gene within the cell, and you edit multiple gene within a cell, and you edit multiple genes within multiple cells. How do they work differently?
How does each one of those work differently?
Yeah.
Um,
What's the challenge that's kind of putting ahead of if you want to progress from single monogenic editing to a multi editing?
yeah.
Yeah.
So I would say it's quite, at least in adult, like in the context of gene therapy in adults, there's no one who does a single gene in a single cell. It's always like either, a single gene in multiple cells or multiple genes in multiple cells. And of course, so far, all the focus has been entirely on single genes in multiple cells. In terms of what makes it more difficult, obviously there's an issue with like, okay, like if you have a certain error rate in your editor, the more edit targets you go after, the more opportunities there are for mistakes. Now, in reality, this kind of varies depending on the editor dose and on some other factors. So, like the scaling laws are not quite as bad as your kind of initial instinct might suggest. But it definitely is more challenging to deal with like off targets and insertion deletion errors and stuff like that when you're going after multiple genes. There's also an issue of like, okay, if you need like this certain level of editor, if you need like an editor dose of like this amount to successfully edit one gene, well then maybe you need twice that amount to edit two genes, and you can't just keep adding editors indefinitely. The cells only have a capacity to break down a certain number of editor proteins, and like there's other kind of bottlenecks that prevent you from inserting infinite editors. So that's dosing, and like cytotoxicity are issues as well. Yeah, I think those are the biggest ones.
Yeah. So I also found your analogy of making analogy of editor and also gene therapy and with compute and also software quite interesting. So you're equating kind of editor to software and gene therapy to editor to compute and gene therapy to software, from your perspective. So I found this quite interesting, how people can approach CRISPR in this way. I think for those who are not really sure about what gene editing is in a CRISPR sense, could you give a brief intro into what base editor and prime editors are, and how they... You talk about prime editor, how it makes single nick in the DNA instead of double strand cut. So I guess for most people, they don't really understand what's going on and how the error rates, it's not gonna affect your expectation of your whole body being ruined through editing. Yeah.
Yeah. I mean, I can explain it and then explain why we don't think the whole body will be ruined via editing. So prime editors are, I mean, I can start with base editors if you want. Base editors, in some ways, more simple. So base editors were created in 2016 at the Lu Lab at the Broad Institute at Harvard. And the whole idea behind base editors was like, there's all these problems downstream of the fact that we break both strands of DNA. So how can we make an editor that works without breaking both strands? And there's these little enzymes called deaminases, which can basically alter a single letter in the genome from let's say from like a cytosine to a uracil. So they chemically alter a single base in the genome. So someone basically had the bright idea to take a CRISPR protein that could find and locate a gene somewhere in the genome and to stick one of these little enzymes on the CRISPR, right? And so the CRISPR floats over it, finds its target, and then once it finds the target, the deaminase enzyme basically latches on to the spot in the genome and it chemically alters the base there. This is kind of a goofy idea, right? It's like you find some car that's going to whatever, a Taylor Swift concert, and you just stick a camera on there to watch the concert yourself, right? It's a little bit like that. Very kind of ad hoc solution. And there's all kinds of problems with it too. Okay, so the deaminase enzymes, they don't actually change one DNA base pair into another. They change one of the two letters into a completely different letter. So changes the cytosine to a uracil. And if you know much about biology, this sounds really strange because uracil's not even normally a base in DNA. Uracil's normally part of RNA, not of DNA. So you're okay, this makes no sense. Why do you want to do this? Why do you want to change this random letter to a completely arbitrary thing? The answer is that the sort of pieces of the cell that make proteins treat uracil like it's a thymine, which is actually a base in the DNA. And also, there's this really funny thing where sometimes most of the time, the cell's repair enzymes will come by, see that there's a uracil, and be like, this doesn't belong here, and it'll repair it and undo the edit that you made. But sometimes it just, for whatever reason, just kind of randomly, it will decide that the uracil is actually correct and it belongs there, and then it will change the letter on the other side to match the uracil to basically form a complementary base. And if that happens, then the edit is actually complete. You've actually changed a base pair in the genome. So, there's some other tricks that they use for base editors. I don't know how deep we want to get into this. I think I'll skip some of the finer details. But base editors come out in 2016. It's this amazing new technology. You can suddenly make edits without breaking both strands of DNA. But the problem is they're quite limited. These deaminase enzymes, they can't just alter any arbitrary base pair into any other base pair. They can only do a couple. In fact, at first, they can only do 2 out of 12 base pair changes. A year or two later, some other people from the Lu Lab made a different base editor, which could alter 2 more of the 12 base pairs. So now we're up to 4 out of 12. And if you combine those 2 that they made in clever ways, you can get up to 6 out of 12. But that's still only half of base pair changes, and a lot of changes require more than one base to be changed. So people are thinking okay, how do we go beyond base editors? How do we make it so we can change multiple letters at once? And this guy named Andrew Anzalone at the Lou Lab has this really clever idea. One day he's like, okay, what if instead of putting like a deaminase enzyme on the the CRISPR protein to like change a base pair chemically, what if we like put a reverse transcriptase? What if we so to back up a little bit to make this all make sense, the way that CRISPR finds and locates a gene is using this thing called a guide RNA. So a guide RNA is like a little piece of RNA that sticks to the CRISPR protein. And it basically acts like, a cop might go look for a criminal by using their picture, right? The cop might look at someone and like look at a picture and be like, do they match, right? A guide RNA functions a lot like a mugshot does for a cop. It basically tells the CRISPR Cas9 protein which gene it's supposed to edit or which gene it's supposed to cut. The way this works is kind of like very interesting. The CRISPR-Cas9 will bond to someplace in the DNA. It will like unravel it, and then it'll check to see does the guide RNA match that section of DNA. If it does, the CRISPR will basically kick into action and like make a cut. So Andrew Anzalone had this really clever idea. He's like, okay, we have this guide RNA. It like matches up with the DNA. What if we extend the guide RNA and use it as a template for the changes we want to make? So basically, like, the guide RNA has some bases in it, right? Andrew's like, why don't we extend the guide RNA and add on the bases that we want to insert into the genome? You're like, okay, so how would this actually work? Andrew's answer is to use another enzyme called a reverse transcriptase. What this does is it turns RNA into DNA. So basically, you have this guide RNA, which serves as a template for the edit. And then you have this other enzyme, which takes the template and turns it into DNA. So it's like a very clever idea, right? Like you basically, you cut the DNA on one side, you pull up a flap, and then you use this guide RNA to like tell you what base pairs to add onto the end of that. So this is how prime editing is born. Basically, Andrew comes up with this clever idea to like use the guide RNA as a template to insert some change that we want to make. And amazingly, there's all kinds of reasons why this shouldn't work, right? Like, first of all, once you add bases onto this flap, you kind of got like two flaps that are like overlapping, right? It's like two pieces of spaghetti that are like competing to like bind to the DNA. And the one that you edited, of course, is not going to bind as well because the bases are different than the other strand. So like you might think that like, okay, there's no way this is going to work, right? Like how did the strands get stuck back together? You just have these two loose flaps like flopping all over the place. But cells are so weird. Like it turns out that if you make the edits on the right flap, the cell's inherent repair mechanisms will cut the other flap, the non-edited one, and they just seem to like doing this. They like to cut one of those flaps in particular. And there's a bunch of other tricks like this that help this process actually work, right? It's just like it's like someone like duct taped an entire car together. That's what prime editors are like, and somehow it works. Yeah. I mean, this is the difference between like computer science, it's kind of like this. People glue together like programs in all kinds of wacky ways. But it still blows my mind that prime editors actually work as well as they do, because it's like it's just a bunch of like really clever hacks all stitched together into like one goofy solution. But it works amazingly.
Yeah. So does it work in a small scale or in the lab only, or does it work in real life?
It works in mice.
How do you make sure something that works in mice that also work in other species?
So the DNA structure is pretty similar. And also we can test to see if prime editors work in human cells. You can culture human cells in the lab, and unsurprisingly, it actually works. And humans and mice are not the only two species this works in. Prime editors have been used in wolves and chimpanzees and all kinds of animals. So it's not too surprising it works in humans. Um, yeah.
So now you have all the standards ready. You have all the research ready. So you guys are here to engineer every component into Bootstrap Bio. So how do you go about it? What's the plan?
Yeah, so good question. Our basic idea is start with an organ that we can easily deliver editors to. So the obvious answer here is the liver, but there's a couple other organs that we could potentially go after. It's also quite easy to get editors to the eye, and there's some other places as well. Um, and what disease do we want to go after? Well, the most the like sort of number one liver disease in the world is non-alcoholic fatty liver disease. Something like a third of all adults have this. Most of them are asymptomatic and undiagnosed. Um, but even among those people, the disease can actually have a reasonably large impact on healthcare expenditure and also on like life expectancy. But NAFLD, non-alcoholic fatty liver disease, it often progresses into much more serious forms of the liver disease, like non-alcoholic steatohepatitis, and in the worst case, cirrhosis, and that liver failure. And that liver failure happens, you can actually die. And if you get cirrhosis or NASH, like your quality of life becomes significantly worse. So we're like, okay, these are obviously pretty serious issues. If we can treat NAFLD, we may be able to actually stop progression into these other more severe forms of liver condition or of liver disease, and we can also, treat this condition, which by itself causes health issues. So, we've basically taken a look at like, okay, which genes are actually involved in this disease, and unsurprisingly, you can find dozens. And if you kind of graph the expected reduction in disease risk from editing these genes, it's very large. It's like over 80%. You might wonder, like, okay, will that actually be enough to treat this condition? And we think it will be because there's a lot of drugs that work quite well for several conditions, and the drugs basically act by replicating the effect of a single protective genetic variant. So a classic example here is Stelara. Stelara works to treat autoimmune conditions by um, decreasing the activity of this particular cytokine called IL-23. IL-23 is like involved in the inflammation response. Some people have a genetic variant of this particular gene that reduces the activity of IL-23. And people with this rare genetic variant, they have significantly decreased risk of inflammatory bowel disease, of plaque psoriasis, and like a couple other autoimmune conditions. So in 2006, Janssen Pharmaceuticals like seeds the results of this genetic study, and they're like, wow, if we could make a drug to replicate the effects of this genetic variant, we can make a lot of money, right? And so they do. They make Stelara, and Stelara now earns them like $10 billion per year. But the genetic variant whose behavior it replicates only decreases the risk of inflammatory bowel disease by about 30%. So we think ours can decrease, the risk by north of 80%. Now there's some other issues that we have. For example, we won't get 100% of the edits we want in 100% of cells. So there'll be some reduced effect from that. But even so, we think that it's actually quite plausible to get a larger reduction in NAFLD than companies like Janssen Pharmaceuticals can get for Stelara. So we think this will actually function not only as like a preventative treatment to like prevent people from getting NAFLD. We also think that it'll function as a treatment itself, much like Stelara functions as a treatment for plaque psoriasis and Crohn's disease.
Right. So let's dial back a bit. So your CSO, Chief Scientist Officer Yuan, so he is the one of the very few who did multi-gene editing, multiple cells.
Right.
So what
Right.
did he do that make you confident in terms of treating liver disease?
Yeah, good question. So he basically invented this really clever technique called CRISPR-Dapperase. So
Mhm.
one of the challenges for gene editing is how do you actually get enough editors into a cell, and how do you make sure that like all the guide RNAs that direct the CRISPR proteins to the different edit sites actually make it in as well as the CRISPR, right? So there's like these two components to a gene editor: the guide RNA and the CRISPR. They both have to come together inside the cell to make that edit. So what Elijah did was he figured out this very clever way to package up a bunch of guide RNAs into a single plasmid. Um, so a plasmid is like a loop of DNA that you can get inside of a cell, and you can encode a lot of stuff in DNA. You can encode the editor protein itself. You can encode the CRISPR-Cas9 protein. You can encode the guide RNAs. You can encode pretty much anything else. But there's challenges to efficiently packing all this stuff into a single plasmid. You can't just like put unlimited DNA into a plasmid and get it inside of a cell. The more DNA you try to fit in, the harder it becomes. Normally, the way that you express something like a guide RNA is you put this thing called a promoter in front of it. So a promoter basically tells the cell, start transcribing a gene here, right? So it's kind of like a landing pad for like your airplane or like your helicopter. Think of it like a helicopter landing pad for like the machinery that turns DNA into proteins. Um, but if you have a promoter for every single guide RNA, it's kind of like having a helicopter landing pad on every single apartment building. It's hard to fit a helicopter landing pad on like every single house, right? It takes up a lot of space. Um, So what Elijah did is he figured out this really clever way to really tightly pack all of the guide RNAs into a single what he calls a DAP array, which is basically like a guide RNA separated by these short sequences of DNAs called tRNAs. And tRNAs are like 75 bases long, which is quite short, frankly. And you can transcribe. How do I put this? This gets a little bit technical, so tell me if this is too hard to understand. But you can basically you manufacture this single really long string containing all the guide RNAs. The string is basically like RNA in this case, and then there's some cellular machinery within like basically every cell that will cut out those tRNAs from this like long string. So basically, it'll chop all the guide RNAs into like their own separate pieces, and then it'll free them from this big string. So I don't know if that. I'm not sure if my explanation was like good enough to make sense. The end result is that you can efficiently pack a large number of guide RNAs into a small length of DNA. Also, tell me if that didn't make sense.
Break into different locations.
Yeah, exactly. It's basically like a cookie cutter. You have like a sheet of like gingerbread, and you have like a cookie cutter, right? And you're like stamping out like gingerbread men from this like cookie sheet. But you want to do it efficiently so that you don't waste a bunch of the cookie sheet. It's kind of like that, but with like with
Okay,
RNA
so
instead.
that basically makes you think it's possible to run multi-gene editing and multi-cells level.
I think it would be possible without this technology to some degree, but this technology just makes it a lot better and makes it a lot more feasible.
Efficient. Right. Yeah. So I guess my next question would be, what are the compliance challenges
Well, yes.
for you guys? I remember in the invest memo, there's a roadmap of saying, so in 2026 and 2027, Bootstrap Bio will begin human trials in Honduras, and we can shave several years of our time to market by following the path blazed by gene therapy company Minicircle. And I checked the Minicircle; it's quite also very interesting. So they developed this gene therapy in Honduran city for muscle enhancement and longevity without actually FDA Food and Drug Administration approval. So I think here's a trick there. Like different countries have different regulatory oversight. So I guess as a biotech company, you guys do have the strategy of leveraging geography in terms of speeding up development.
Yeah. So there's not really any regulatory roadblocks when it comes to R&D. Thankfully, we're able to basically do our lab experiments, without really any compliance issues. But it does become trickier when you have to deal with, human trials. Um, we'd like to eventually get approved in the United States, but the problem is if we go straight for FDA trials, it's going to, we've heard quotes from people in the industry who said like you should budget like 400 to 600 million dollars for an FDA trial. Like, if we have to budget $500 million for FDA trials, like this, it's very hard to see this actually happening because it's a new technology. It's like a new idea for how to treat disease. If we have to pay $500 million, no one's going to fund it because there's uncertainty that it will work, right? It's a whole new approach. Um, so, I mean, this is part of, I guess, a broader set of complaints about the FDA. But like the clinical trial process has become so expensive that it's very hard to innovate in the U.S. Um, so we started looking into other countries where we might be able to run clinical trials, and it seemed like one of the most obvious potential solutions for us was this special economic zone called Prospera in Honduras. Um, uh, and again, there's this gene therapy company there called Minicircle, which has done their own clinical trials. So we figured like, okay, like we can get this done in Prospera in like probably under a year. At least we can go through like most other trials in under a year. Um, we can save hundreds of millions of dollars. It just kind of seemed like a no-brainer. And like if it works, if we pass clinical trials in Prospera, of course we won't be immediately approved in the U.S. But if we have a cure for, like fatty liver disease or another serious liver condition, like there's direct flights to Roatan Island where Prospera is, from Miami and from Houston. So like people can just fly from the U.S. to get treatment. I mean, they already do this a lot for other medical treatment. So like if we have a cure for a dangerous disease, like they're gonna fly.
Well, I think Balaji is one of your investors or very early
Yeah,
investors, and he is pretty big in the proposing the network state
yeah,
in the industry of cryptocurrency and blockchain. So I think what you just mentioned about people flying over to another island for medical treatment, it's quite interesting and kind of match to what his ethos are. Like in the future, they'll kind of humans will be more nomadic, break the barrier of sedimentation, and the world will become more fluid in terms of technologies. I guess we met in a crypto meetup, and Minicircle
yeah.
seems to be a very interesting example here because it only accepts crypto payments. Um,
Oh, interesting. Huh.
yeah, so I did some search today online. They only accept cryptocurrency transactions, I think. Um, yeah, so their transactions are based strictly on cryptocurrencies. So it's quite interesting to bypass this country barrier through more private money. So obviously you are also touch on this in blockchain industry. So I just want to hear about your perspective in terms of merging biotechnology and crypto together.
Yeah, interesting. I didn't know that about Minicircle, but that's cool. I'm always kind of on the lookout for ways that we can incorporate new technologies into our roadmap that would be beneficial. I don't know if we would restrict our therapy to only be payable in cryptocurrency. I think if that poses additional barriers on customers, then I think we probably wouldn't choose to do it. Um, but be
Right.
open to taking payment.
Yeah.
We'd,
So I
would certainly
guess
be open to
crypto really
taking payment in cryptocurrency.
Mhm.
Sorry, go ahead.
Yeah. So it's more of a borderless concept of democratizing treatments to people who doesn't really have a passport advantage. I guess I can see the connection here for crypto and biotech treatment. Not sure what's your thoughts are about basically biotechnology being restricted by countries like the US, and corporations just too slow in terms of developing into the inertia of monogenic editing.
Yes.
Whereas we need disruptive biotech startups to go into the space and engineer the already existent research components together. However, to break that, you need some tricks. You might need to fly over to a different place in order to speed up the process. So what's your take of corporations and startups in the future, in the realm of biotechnology? And also
Pretty broad question.
Yeah.
Yeah, so I mean, we definitely want to spread out to like multiple countries and make this broadly available. I mean, it would be a shame if you can only get treatment because you're in country. I think that that sucks. And obviously, from a business point of view, it's also stupid because the more customers you have, the better. In terms of corporations and the rate of innovation, I think that there's definitely, like, you definitely make a good point. There's a lot of uh, just very conservative, like uncreative thinking when it comes to like how to actually treat disease. There's really no good reason why no one has done multiplex gene editing for like treating these conditions before. Climate editors came out in 2019. At any point during that 5-year window, someone could have chosen to do this, and they haven't. Well, why? I mean, there's no good reason for it. There's a lot of it comes back down to like biologist views of like the genome must be super complicated and you don't know what's going to happen, which is basically just wrong. Like, we do know how it works, we do know which genes to edit, but this very conservative mindset dominates in biotech, and there's some good reasons for it and there's some bad reasons for it, but a big part of why biotech is so conservative comes back down to the regulatory environment in the US. If your clinical trial costs $500 million to get through, companies are only going to want to take safe bets. They only want to bet on something that's like almost guaranteed to work. Because if they fail and they get all the way to like phase 3 clinical trials, it's so expensive. I mean, each failure is like $500 million failure. So, your rate of innovation just slows down because innovating is so much more expensive. So I think the regulatory piece is actually a really key part of the innovation pipeline. If the regulatory process is cheaper, you will get more innovation just almost inevitably.
Yeah. So, but there are still many successful biotech startups in the country.
Right.
Do you have any successful biotech startups strategies you really like and would like to incorporate into your company? Do you admire any the startups out there?
Yeah, let's see. It's funny, like having a lot of the startups that I admire the most are actually not in biotech. Like SpaceX is, I think, one of the coolest companies to develop new products in a long time. I think in the biotech field in particular, I think Genentech's story is like super cool. This whole idea of like creating insulin in like bacteria was like this kind of radical concept when it was first proposed. But it's like so clever and like it works so well for so many different applications. You basically turn those like bacterial plants into like manufacturing plants for arbitrary chemicals or like proteins. Minisurgical, frankly, is also pretty cool. I think that what they're doing is really neat. And I hope that they continue to innovate and push the envelope. And I also like they're doing some other interesting things, like this whole idea that like we can only ever treat disease, and like we can't ever hope to like we can only ever raise the floor of the human experience, but we can't ever raise the ceiling. I think this is just fundamentally misguided. You want not just the worst experiences of people's life to be less bad, but the best experiences to be better. And there's no reason biotech can't fundamentally provide that. It really can. It can. And many circles, uh, basically, I think like leading the way on that. The follistatin therapy, of course, can be useful to combat aging, but frankly, it can also increase muscle strength in people who are not old. So I think I like their approach and I like their philosophy.
Yeah, so that takes us back to the very initial question I asked about plus minus intelligence, why people are so resistant to plus intelligence as opposed to minus
Yeah.
intelligence. So you're saying is when we reduce the cost of gene editing for intelligence to where everyone can afford, then it will be a norm. In your case, as a CEO, what's your strategy of reducing the cost of your therapy?
Yeah, really good question. So in the context of our therapies, it's hard to answer for brain stuff because again, delivery is not really solved at this point. When we have examined this question, a big piece of our thinking is like, what is the marginal cost per dose of a treatment that we create, and like, how do we pick technologies that will minimize that cost? I mentioned earlier that there was a company called Dino Therapeutics which made this like viral delivery vector called an AAV capsid that can actually deliver to the brain right now. But in addition to sort of the limitations with the amount of genetic material that you could pack into each AAV, and the limited delivery efficiency, it's also just extremely expensive. I think we calculated that for delivering to the brain, the whole brain, the cost of the delivery capsule alone would be like over $1 million. And if your cost per dose is that high, if your manufacturing cost alone is that high, you can't ever create an affordable therapy. It's just impossible. So in the long run, when we try to solve delivery, we really want to think a lot about cost and how we minimize it. From a manufacturing perspective in particular. Obviously, a lot of drugs and treatments are most of the cost is R&D, and the manufacturing is actually often cheap. But if the manufacturing is not cheap, then there's no way you can reduce cost.
Right. So what's the barrier for manufacturing cost?
So it depends what delivery vector you're talking about. For like liver treatment, there's not much of a barrier. You can manufacture treatments for liver at scale quite cheaply, for like less than $1,000 per dose. I think we're likely to be able to get to less than $1,000 per dose, or per treatment. For brand stuff, I mean, the answer is just like whatever technology you use, you have to find ways to manufacture it cheaply. And I don't really have any concrete insights there because again, there's plausible methods to get gene editors into the brain, but they're all just ideas at this point. There's no like proven, very easily usable off the shelf technology for brain delivery.
So what do you, as an innovator, like when you wake up every day, what do you fear the most?
By far, the thing I fear the most is AI. It sounds like a silly question, but or it's a silly answer, but it's honestly the truth. I mean, everything depends on this. What happens with this technology. Are we going to be able to control it? If so, who controls it? Um, is it better for like? Is the technology itself? Does it lend itself better to warfare and like offensive, aggressive things, or does it lend itself better to defense and like, protecting people? I don't think anyone really knows the answer to that question, but the answer will become very important. And then there's other kinds of questions like, okay, what is the point of people if AI becomes smarter and better than us at everything? This sounds like a silly thing for a biotech founder to think about, but actually, it's in the long term, it's quite relevant because it has implications for how useful the technology that we're working on ultimately ends up becoming. There's other kind of nearer term concerns that I think about, like, how do we make sure that we, like, what's our research strategy for the next month? How do we make sure we get these plasmids delivered in time so that we're not like bottlenecked on having plasmids available for experiments? Kind of more mundane stuff like that. How do we hire the best people? How do we like close our next round? Stuff like that. So on a day-to-day basis, that's what I'm thinking about. Long term, man, it's hard to find anything to be more concerned about than AI. Maybe nuclear war.
Yeah, exactly. So my last question for you is, like, in terms of the roadmap, the schedules, to me, they are quite tight. So what's your, how did you make these numbers? How did you make these plans?
So they're inherently they're speculative to some degree because there's a lot of unknowns represented in those timelines. For example, how well did the experiments go? How many iterations do we have to go through before we find like the best technique? How are we able to do clinical trials in the timelines that have been told to us, or like those optimistic timelines? We kind of assume that they're a little bit optimistic. Maybe they're like really optimistic. Maybe they're realistic, in which case it would be shorter. What else? So we think we're probably going to have our first editing results in like maybe 2 to 4 months, like our first big results from the editing. And at that point, we'll be able to show like, okay, what edits can we actually make in liver cells? Like which genes were we able to edit? What was the editing efficiency? Kind of those basic metrics. And at that point, like if those results come back positive, like, oh my God, this is going to be a very, very interesting company. Because if we can make one treatment for just liver disease, the market for that is like 50 billion just in the US. Right? And like we're a tiny startup. If we can make 50 billion in revenue from the treatment we make, that is going to be a extremely, extremely valuable investment. Of course, there's a lot of uncertainty, and we don't know what's going to work. Just like any startup, we have a decent chance of failure. But I don't think people understand how big the opportunity is here. Maybe it's not us. Maybe we fail because we can't execute, or something else goes wrong. But like someone is eventually going to figure this out. The question is just who and when. I think if you wait for big pharma companies to try to solve multiplex gene therapy, it's going to take another 5 years. And when they do, they're not going to be concerned about raising the ceiling of the human experience. They're only going to be thinking about how do we make the most revenue for our bottom line using the most conservative possible strategy. Mm-hmm. So I see that the good that Bootstrap Bio can do is to basically speed up the development of this technology and also expand people's conception of what biotech is actually good for. I don't think it's enough to just say, we're going to make you not sick. We want to make people's lives actually better.